like clinical state was still studies. Yes, a bit of is like just
A lot of people emailing me that they're ill, and I'd say the number of people here confirms that. So look after yourself, get your sleep, stuff like that. And you can tell from my voice, I too had whatever this is, other than COVID. So last time we were talking about how after the number of neurons is pretty much set in the nervous system, that there's a period of an exuberance of synapses followed by a time when many synapses are lost and others are gained, a period of synaptic rearrangement. And this is very extensive and to some extent will go on the rest of your life. And what we also talked about is how the decision about which synapses will come and which synapses will go is usually dependent on activity, synaptic activity, which is to say, as Hebb suggested, that successful synapses stay and unsuccessful synapses go away. And so we're going to explore that some more and take up, in particular, in the visual system. And we'll keep returning the visual system several times, as we have already. And I want to point out to you what it is about the visual system that makes it such an important model and so useful. Basically, it is that there are these two-dimensional arrays of receptors that are reflecting the space around us. And it helps that there's two of them. So you will see sometimes you can manipulate one and not the other, and that's useful. And it helps that they're sticking out here in the periphery where it's relatively easy to manipulate them by, for example, putting tetrodotoxin in an eye to stop action potentials and stuff like that. So never worked on visual system myself, but I think I don't think I'm making this up. I think many neuroscientists have vision envy, right? Because the visual scientists seem to be doing so many cool things. And we'll see some of those here. And so as we take up the visual system, the first thing we need to do is review the anatomy of it. And to some extent, you should have seen this already, but I want to make sure that we're all starting in the same place. So as you know, that double convex lens in the front of your eye, plus the shape of the cornea, means that the image of the outside world is projected upside down and backwards onto the back of the eye. Just like when you played around with a magnifying glass and you held it out a long enough distance, the scene that you'd see would come into focus, and it too was upside down and backward. And I want to make sure that we all know how the visual scene projects onto the retina and from the retina to the world. The first thing I have to deal with is sometimes people come with the mistaken idea that the right eye projects to the left brain and the left eye projects to the right brain, and that's not true, right? Wouldn't that be simple? But it wouldn't be effective. And in fact, the thing to remember is that the left visual field, everything you can see while you hold your eyes still, the left visual field projects to the right brain and the right visual field projects to the left brain. But both fields use both eyes to do that projection. So light always travels in a straight line. So you can think for yourself what would happen if there's something in your right visual field. Light bouncing off that object will reach the nasal half of your right eye and the temporal half of your left eye. Temporal as in close to the temple. Easy to remember. So the right visual field is going to project to the nasal half of the left retina and the temporal half of the nasal half of the right retina and the temporal half of the left retina, and vice versa. And so you end up with a situation where those retinal ganglion cells in the left eye that are being stimulated by the right visual field, they will not send their axons across the midline at the optic chiasm. So they'll continue to go to the left side of the brain so that you end up with the right visual field projecting the left side of the brain. And again, vice versa. How's that so far? All a review, right? So I'm going over it pretty quickly. And in mammals, at least, the majority of those retinal ganglion cells send their axons to a particular portion of the thalamus known as the lateral geniculate nucleus. And the lateral geniculate nucleus, and just in case we use these terms, we refer to the part of the bundle of axons before the optic chiasm as the optic nerve as it's sticking in the periphery. But then once it crosses the midline and enters the central nervous system, there our convention is we call a bundle of axons a tract. So it changes from being the optic nerve to the optic tract, but it's the same axons. And some will project to the midbrain, the superior colliculus, but the majority in mammals will project to the lateral geniculate nucleus. The lateral geniculate nucleus, in turn, will send its axons to the primary visual cortex, which is in occipital lobe. And in humans, most of the primary visual cortex is tucked between the two hemispheres, tucked away in here. And we call that primary visual cortex V1. I'll review. Everyone clear on that? Okay. So, what we're going to see is that a big decision that has to be made in putting together a visual system is at what point do you keep the inputs from the two eyes separate, and at what point do you bring the input from the two eyes together? And what we're going to see is before birth, before the eyes are even open, before there is any visual experience per se, before that time, spontaneous activity is going to serve to segregate the two inputs, keep the inputs for each part of the relay system either getting from one eye or two eyes. So when we say there's only input from one eye to a region, we say it's monocular. And when we say it's getting input from two eyes, we say it's binocular. And in the lateral geniculate nucleus, remember these axons are fresh from the eye, right? It turns out that the inputs from the left and right eye are segregated. So the layers are numbered. And it's called the geniculate nucleus, right? Because in cross-section, it looks like a knee, like you're bowing, genuflecting, same Latin root for genu. And if we look in these sections, there's a nomenclature for numbering them, etc. I don't need you to know that, but I do want you to know. I don't need you to know which ones are getting from the ipsilateral eye and contralateral eye, but I do need you to know that they're segregated. So in this case, the left lateral geniculate nucleus, some parts of the geniculate nucleus, one layer is getting input exclusively from the contralateral eye. Right underneath it is a layer of neurons that are getting input from the ipsilateral eye, etc. And as we'll see, that segregation of inputs from the two eyes is going to be important. If we take the next step, this is just reviewing what I've told you. The next step is these lateral geniculate nucleus neurons, they're all going to send their axons to the primary visual cortex. And the general rule of cerebral cortex, neocortex, as it happens in us mammals, etc., is that all synaptic input always arrives in layer 4, the middle of those 6 layers. And you can take that to the bank. No matter which part of the cortex you're looking at, input from outside that area is going to come into layer 4 and synapse on neurons in layer 4. So there's the first synapse from the LGN to the cortex. Everyone have that? And then the other rule about cerebral cortex, the general rule that, kind of brings to mind that maybe it's just a generalized problem-solving machine, is that within any column of cerebral cortex, there are lots of axons and synapses going up and down the column. And the columns tend to be about 0.5 millimeter wide. So in any one unit of cortex, you can think of this column of cortex, peel surface at the top, white matter at the bottom. where all the inputs are coming exclusively to neurons in layer 4, and then the neurons in layer 4 in turn synapse up and down that column with all the other neurons in that column. You know, the neurons in layer 6, 5, 3, 2, 1. Right? And the output of that column is also very segregated. Output is almost exclusively coming from layers 3 and 5. That's not a part of our story here, because in looking at visual cortex, we want to ask ourselves, in layer 4, are the inputs from the lateral geniculate nucleus, which remember are segregated by either the ipsilateral eye or contralateral eye, do they continue to be segregated by eye in layer 4? And the answer is, it depends on when you look. So, questions about that? So in the adult, in the adult at least, the input to layer 4 is segregated. It is monocular. Each little region of layer 4 is either getting input from the ipsilateral eye or the contralateral eye. And that means, as you might have guessed, each of those columns, right, is initially getting information from one eye or the other eye. And it turns out that we can see these ocular dominance columns in a cool experiment where someone you might have heard of, Simon LeVay. If you've heard of Simon LeVay in terms of, you know, the difference between gay and straight men and the hypothalamus, etc., he was famous for that long before that ever happened. He was famous for these studies I'm going to tell you about, where he took radioactively labeled amino acids and injected them into the eye, where they get taken up by retinal ganglion cells in the retina, and then transported to the lateral geniculate nucleus, where some of the products of those amino acids would end up in that cell, and it would then send radioactively labeled proteins to the end of their axons. So this is so-called transsynaptic transport of a label. So he would then kill the animals, take out the visual cortex, slice it real thin, put it on photographic film to see where the input was going. And in adult animals, when you do this experiment, he would end up seeing, you know, that each lateral geniculate nucleus was getting a particular input from a particular eye. But more importantly, when he laid out the visual cortex flat and did a slice across, whenever his section was in layer 4, he saw this beautiful segregation. The silver grains here represent photographic silver grains from the injected eye. And lo and behold, these bands, which you can see, they don't look like a street grid, right? They're kind of snaky and eeling around one another. But these bands also tend to be about 0.5 millimeter in width, meaning that with the right contrast, you'd be able to see them with your naked eye pretty easily. And so here is this pattern where cells in layer 4 of the adult visual cortex are receiving monocular input. So far, so good. And that means that certain bands are getting input from one layer of the lateral geniculate nucleus, and the band nearby is getting input from a different layer of the lateral geniculate nucleus. The interesting thing that happened, and what made Simon famous, was when he did the same experiment in kittens instead of adult cats, because he got very different results. So what he saw is that when he did that experiment in kittens, no matter which eye he injected, there was a totally homogeneous, even innervation of layer 4 of visual cortex. And what he saw is that once the animals open their eyes, the inputs to layer 4 tends to start segregating. And that suggested that maybe visual experience was important for segregating ocular input to the visual cortex. However, later people would show that the same thing is happening in monkeys, and in monkeys, the segregation of input to layer 4 happens before birth, before the eyes have seen anything. And so the question arose, what's driving this segregation of input to layer 4? And the answer was activity. Oh, and here are the experiments, and this happens to be in cats, I think. So here's one preparation where he injected the eye, and you can see all of layer 4 is getting input from that eye. And if we looked in slightly older animals after the eyes have opened, now you start to see that it's a little spotty, right? Some areas are getting a lot of input from the injected eye, and some are not so much. By day 39, things have really kind of sharpened and come into focus. Now there's very little overlap, and by the time you look at an adult, you see in this cross section, you're seeing different bands. And if we sliced the layer 4 horizontally instead of cross section, we'd see these bands are those snaky lines that you saw before, those ocular dominance columns. Okay. And this segregation of input is not due to visual experience, it's due to spontaneous activity. So whenever you make a neuron, whenever a neuron arises, once it starts expressing all those genes to make it a neuron, the stuff for the machinery for action potentials and stuff like that, every once in a while they fire. We have this notion maybe that most of the time, most of the neurons in your brain are quiet because they're not doing their job. But if you've ever stuck electrodes into a living brain, what you know is cells are firing all the time. And the big difference between a cell firing in terms of fulfilling its function isn't so much when it ever fires, but rather it fires more when it's fulfilling some particular function. And probably it's true that no neuron ever sits quiet for more than a few seconds at a time. Okay? And the same is true during development. Once they start arising, they are going to have what we call spontaneous activity, because as far as we know, we don't know what triggers it. Could be just stochastic process, just random noise. And what's interesting about the spontaneous activity of retinal ganglion cells in the axons in the retina before birth is that they are synchronous. It turns out before birth, the retinal ganglion cells are making gap junctions with one another, electrical synapses, so that if one cell happens to fire for whatever reason, it often triggers its neighbor to fire as well. And so as people look at preparations of the retina flattened out with the spontaneous activity, there are these waves of spontaneous activity going back and forth across the retina. Everyone see that? Imagine a stadium where there's not just one wave going around in a circle, but there are various waves going back and forth, and they come and go and ebb and surge and stuff like that. So far, so good. One consequence of that pattern, that spontaneous activity, is that retinal ganglion cells who happen to be close to each other on the retina are more likely to fire at the same time than retinal ganglion cells that are far away from each other on the retina. Not because of visual input, but because of spontaneous activity. Right? And so this sets the stage for a Hebbian synapse type situation to take place, where imagine we're looking at the left eye and there's a group of retinal ganglion cells that have sent their input to layer 4, and they are more likely to fire at the same time. And therefore, their synapses should get stronger. In the other eye, those cells also fire at the same time. But if at the outset there happens to be more input from one eye than the other, then just as Hebbian synapses are supposed to work. The rich are going to get richer and the poor are going to disappear. Strong, effective synapses become more effective and ineffective synapses become less effective. And with time, that means in any one region of layer 4, I don't know what I have there, in any one region of layer 4, eventually one eye or the other will come to predominate, even though there's been no visual experience yet. Notice that the gap junctions between the retinal ganglion cells and those waves of spontaneous activity, that's probably not a coincidence. Notice that mechanism already starts to impose some order on the neural circuitry. Even though the activity is spontaneous, by having neighboring neurons, and remember, this is a two-dimensional sensory array, right? By having neighboring neurons fired about the same time, now you start to impose order on the way they're innervating the brain. And of course, that only works because long before mammals existed and had their particular visual system working, long before Hebbian-type synapses came to arise. Yeah, just.
How come it's in layers and it's not like cell by cell, it's like different innervated by the same?
Yeah, so, I mean, no one knows the total answer, but you can imagine that what's going on is that there are various domains where one happens to have a slightly higher input at the outset than the other, and that it spreads like that. Does that help? Why isn't it more punctate? Is your why are they in bands instead of not being little points, right? Yeah, it's a great question, and that's because that same process is going to mean, oh, here's a patch of retina. These cells that are really close by, they almost always fire at the same time. They're gonna, and you know what? Then there's the area next to them where they fire almost as often as those, and so they'll start to take over, etc. And so you end up with a situation. And so the magic question is, why are they half a millimeter wide? Why aren't they a millimeter wide? And I don't know. I don't think anyone knows. But presumably, it just has to do with the rules of how this synaptic strengthening is going on.
There's no like genetic influence that's like kind of has more connections to a certain layer, and then there's like a refinement with.
At this stage, people have spent a lot of time analyzing those ocular dominance bands, and nobody's been able to find a pattern that explains them, right? They seem, at that stage at least, they seem more or less random in terms of which eye they're getting input from. But in a moment, we're going to take in the next chapter, we're going to take another step forward, and there we're going to see that, yeah, for this system to work, it has to be the axon got to the approximately correct part of the cortex in the first place, guided there by things that got nothing to do with spontaneous activity, i.e., right, adhesive molecules, ephrins, stuff like that. And a cool experiment that I like to talk about showed this in ferrets, where they weren't looking now at the cerebral cortex, but they were looking at the lateral geniculate nucleus. So the pattern of monocular input, the lateral geniculate nucleus in ferrets, is different than in the other animals we talked about so far, okay? Different than in primates. And so here are cross sections of the lateral geniculate nucleus. And in control, and the other reason people like to study ferrets is because they're born at a very altricial stage. That is, there's going to be a lot of development happening after they're born. And so the experimenter can get access to them and do things. In the adult ferret, this is what the pattern of input to the lateral geniculate nucleus looks like. So here, the pseudocolors, red is coming from one eye and green is coming from the other. And I forget which is which. And so this is what things look, that is, so the right lateral geniculate nucleus is getting input from the green eye in this area, and the left lateral geniculate nucleus is getting input from the red eye in this area, whereas the rest of the area is getting input from the other eye. And so what happened in this experiment is they started injecting drug to get rid of the gap junctions in the eye. And if they got rid of gap junctions in both eyes, notice the segregation didn't take place. In fact, it kind of looks pretty much like the state of affairs at postnatal day 1, where there's less focusing of the inputs to the two eyes. And in another case, if they injected the drug only into the red eye, notice that the green eye more or less took over. So this is exactly what you'd expect if there's some sort of competition going on between these animals, Hebbian-like synaptic competition going on, and that the waves of spontaneous activity brought by the gap junctions is important for that segregation to take place, which is the first step towards segregating, towards the next step of getting input to the right part of the visual cortex at the right time. Well, I like to show this kind of classic illustration to point out to you that this is what the cortex looks like in a newborn, 3-month-old, 2-year-old. This proliferation of dendrites is showing you also a proliferation of synapses. The number of synapses that's happening in the postnatal human brain is, pretty astronomical, huge. And people have tried to quantify this in other species. So here, the human cortex, this is looking at the total number of synapses, and it's a logarithmic scale you can see here. And so what you're seeing is in the first few months of life, there's an incredible proliferation of synapses. And what's going to happen after, somewhere in the first year, is the total number of synapses is going to go down. And you won't be surprised to learn that it probably matters which synapses come and which synapses go. And you can see the output of that as you watch the baby's behavior get more and more complex and have it be more and more behaviorally competent. People have also looked at it in cats, and it doesn't matter which part of the cortex you look at, it's pretty much the same. That is, there's a period of exuberance followed by a decline in the total number, whether you're looking at auditory cortex, visual cortex, prefrontal cortex. Or for another example, you can look at in humans, where they're keeping track of a particular kinds of neurons in 2 different layers of the cortex. And it turns out that the peak in the number of dendritic spines happens, before age 10, and then for the rest of your life, you start losing them. Once you hit 40, basically brain weight goes down by about, as I recall, about 5% per decade. Right? Less and less. You got to get by more and more with less and less. And another way of looking at this process is to look at at MRI images of the cortex, where you're looking at instances, and here they have used pseudocolor to show you how thin the cortex is. Because this net loss of synapses across development is, in structural terms in the cortex, is seen as the gray matter getting thinner, and of course the white matter's getting thicker as myelination takes place, etc. And here we're looking at age in this axis, and the pseudocolor is red is where the cortex is thinning fastest, and blue is where the cortex isn't thinning. And the important thing to note is there are some parts of the brain that are still maturing even at 20 years of age. And here we're just, yeah. And there's a sex difference in the process. In general, male brains tend to be heavier than female brains, but that's also because male bodies tend to be bigger than female bodies. How's that? And what you know from your other neuroscience classes is that so the last to mature is the prefrontal cortex. And as you know from other classes, the prefrontal cortex, which is far and away which is much bigger in our species than any other primates, that region can be, roughly said to be an area that inhibits behavior. It's the part of the brain that probably says, oh, you felt like doing something, and then the prefrontal cortex said, wait a minute, that might not be a good idea. Right? And as homespun as that sounds, that can explain an awful lot of neuroscience research. And here's an interesting thing to look at. So here we're looking at reaction time based on age on large samples. And so I need to tell you a reaction time, light comes on and you have to hit a switch. And how quickly do you hit the switch afterwards? Or a light comes on and you have to push a pedal with your foot, and how quickly does it happen afterwards? You too are on the downward slope. I'm sorry, your reaction time is never going to be better than it is now. You're going to get slower and slower, and there's nothing you can do about it. What's interesting, to me at least, is here we're looking at the number of driver fatalities, and it is normalized for number of miles driven. Okay, so number of fatalities per 100 million miles. And here it's segregated by age. And notice that 80-year-olds have a lot more accidents per mile driven. Anyone surprised about that? Paradoxically, though, in fact, in 80-year-old drivers in general have fewer accidents than people at other ages. Why? An 80-year-old this year is probably going to have fewer accidents than a 60-year-old this year. Why?
They use exactly.
They don't drive as much. Yeah. But if you standardize per miles driven, they really shouldn't be out there. Probably. But here's the more interesting thing. Which of all these ages, 16-year-olds have the highest fatality rate, and yet they're the ones with the fastest reaction time. So the fact that they have more accidents has got nothing to do with whether they can react fast enough. Because they're even faster than you are. Sorry, you're already past your prime.
Right.
Questions about that? And many people think it could be because their prefrontal cortex is still maturing at this point. They haven't really learned. Right? They haven't had enough accidents. They haven't learned how fragile and horribly mortal they are yet. Stone, come.
Wait, question. I'm curious to get about the ball. Why do you think, I guess the judge gauge gets to see rather than,
Yeah, why is the driving age at 16? That's a great question. Because you can see right there, 16-year-olds are significantly more often to have a fatal accident than 17-year-olds. That's a big jump there. You could argue about 17 to 18. So how come the driving age is at 16? Yeah.
Make it easier on their parents.
Yeah, because the parents want to stop driving the kids around all the time. That's of course, the kids are eager to drive too, but politically, nobody listens to 16-year-olds because they don't vote. But their parents vote, and they want to stop chauffeuring them around after.
What if the driving age was 18, maybe 18 would spike because 16 is so high because
they're new drivers. Good point, right? So they haven't been doing it very long. So that's definitely going to happen too. I agree. A similar thing is how come the drinking age is 21? So I've got a personal history there. No, it's not what you think. I was just the right age that when I went to college, the age of drinking went to 18. So you're looking at someone who has never illegally had alcohol. right? Because I was too much of a goody two-shoes when I was in high school. And by the time I got to college, I can drink, it's legal, right? And that was in the state of Connecticut. The drinking age there now is 21. Well, I understand the political history of that. They lowered the drinking age to 18 during the Vietnam War when they were conscripting 18-year-olds and saying, you're old enough to go die for your country. And people started asking, they're old enough to die for a country, but they don't get to drink and they don't get to vote. And so that was when the voting age went to 18, and that was when the drinking age went to 18. Guess what happened? Then the number of drunk driving fatalities zoomed up. So eventually, politically it was, well, okay, we won't let them, if we let them drink, more people will die. So even though we're still, I guess, and if the draft ever comes back, I think you could, and if they again draft 18-year-olds, I think you can expect the drinking age. So why is the age 16 for driving like all other of these age limit things? It's all a balance of political forces. Questions about that? Yeah. They're reminding me of something. Oh, and note that whatever age you pick, we all know there's some 16-year-olds that are a lot more mature than some 21-year-old, right? And vice versa. So anytime you use an age limit and you choose a threshold like that, it's not going to be the right threshold for everybody, but you got to make a decision somewhere. Okay. Well, I also want to point out to you that we think of myelination. So the myelination, right, is almost exclusively postnatal in humans. And there's lots of myelination going on well into into our 20s. And it turns out that myelination process, you might have thought that it was pretty simple, but in fact, it too could be affected by activity. So not necessarily sensory activity, but activity. So oligodendrocytes, remember they're called oligodendrocytes because they have only a few branches, and each branch goes out and wraps around a particular stretch of axon, and how thick that myelination is due to what? What's signaling from the axon? Neuroregulin, right? So the more neuroregulin the neuron puts in its axon, the thicker the myelination sheath will be. But people have shown that if you use tetanus toxin to reduce action potentials, that the number of the thickness of myelination goes down. This in animal models, of course. So who would have thought that neural activity isn't just affecting synapses, it's also affecting myelination. And you can imagine that might be important too, right? You can imagine that certain pathways, it's really important for the message to get there quick. And so you want the myelination sheath to be thick because that, saltatory conduction and all that. Had enough? Okay. Let's Kahoot. So once the stage is set that activity can decide which synapses come and go, and we can see that sometimes spontaneous activity can itself impose order onto the developing nervous system. But once that mechanism is present, once Hebbian synapses are out there, sooner or later, there's going to be situations where the activity that's driving the neuron to fire is related to the outside world. Once sensory systems start coming online, then we have the chance for the sensory world to impose additional order on the nervous system. And what we're going to see is that for every sensory modality that's been looked at so far, not only is it that sensory experience can mold the nervous system, but in fact, it must mold the nervous system for it to work properly. So that's where we're going. And I have to, this is not George Stratton. This happened later, but at the turn of the century, no, the other century, believe it or not, philosophers did worry about, once they found out that the visual scene was being projected upside down and backwards on the retina, people really did kind of worry about, well, how come we don't see the world that way, right? The answer being, we do see the world that way. That's exactly how we see it. But, right, it is happening that way, but something happens. And so Stratton was, I guess an early student of psychology, but it was also back when psychology was in philosophy departments. And he wondered if he could reverse that by putting prisms in front of his eyes, turn the world right upside down and backwards again, and project, that image onto his retina. And he experimented some with having it on both eyes, but he could never get it right? Everything, he could never make sense of the world at all that way. So he just went with putting a reversing prism on one eye, and he was very careful to wear an eye patch on the other eye. And he only, and wore an eye patch at night. So he went several days where he could see only with one eye, and the world was reversed and backwards, upside down and backwards. And he wanted to know how he could get along in a world like that. And of course, it was pretty hard at first, but eventually, and he did two different experiments. One, he did it for 3 days. And by the end of that time, he was pretty good at taking care of everyday life, right? He could open doors. He would reach out with the right hand to open a door, with the correct hand to open a door. He could pour a glass of milk, stuff like that. Although, he said in the early days it was pretty haphazard, pretty, hit and miss as to how well he did. And then when he took off the prism, right away he felt well oriented. Then he repeated the experiment, and this time he kept it on for much longer. And then interesting things started to happen. One was that he could do things, and he felt like his proficiency at taking care of tasks was about the same as it ever was. What changed for him was his experience of where his body was. And so if you read those, his accounts, it was, really pretty strange, where, looking at a fireplace, he felt like he was looking at the fireplace, everything was right about it, except he was looking with his head between his legs, stuff like that. So his experience wasn't that anything was wrong with the visual field or visual information. His experience was that his body was in a strange way. That's how powerful vision is as a stimulus, right? That he was, his experience was it must be his body's twisted in some strange shape. And the other fascinating thing is when he took the glasses off, it took him the glass off the eye. It took him several hours to be able to do anything again. You'd think he'd be relieved and he'd instantly have his ability to use visual experience right away, but he didn't. As a neuroscientist, what's your interpretation of how Stratton was able to make sense of this visual information that had been reversed? What do you think happened? How many of you think he rewired connections between his eyes and his lateral genicular nucleus? No taker? Thank you. You want to argue for it, or you? Okay. Or between the lateral genicular nucleus and the cerebral cortex, visual cortex? Yeah, maybe. How many think it might have been in terms of getting information from visual cortex and getting it to the rest of the brain and making use of it? Yeah, that seems most likely. And after, rewiring, you can imagine, well, especially in that 8-day experiment, there might have been time for rewiring. Nobody knew how plastic the nervous system was then, right? We didn't even have the name synapse, right? So, but the fact that when he took it off, he could recover in a couple of hours, that's asking a lot to think there's a lot of reinnervation or changing in synaptic connectivity. Yeah.
For the experiment where they clipped the frog's eye, did they keep him alive for like 8 days?
Hold on. I'll get there. Don't steal my thunder. Yeah. Yes. Months. Most people, the interpretation is it must be that higher centers of the brain were somehow able to reorient that information and make use of it and recalibrate his movement so they could get around in the world. I mean, at the 8-day experiment, I guess he was riding his bicycle around town with the world like that. And there's no record that he was hit by a car or anything.
You said that when he was looking at the fine points and stuff, that he felt like he was looking through his legs. So did he feel like he was still looking at everything upside down, or did he just get better at like what?
As he described it, he didn't think that there was anything wrong with the visual field. I mean, by the end of that time, it looked right to him. Okay. But his only way of explaining the orientation was that somehow he was, that he had twisted his body around. In some ways, if you read some of the descriptions, it's like you can't do that, right? I mean, nobody can do that. Well, anyways, just to get to Justin, what I love about this is he's wearing a suit and tie, but I guess people did in college those days. Anyway, so this experience, what Stratton was doing at the Prism, is very much like the experiments we've talked about before, where Sperry took the eye of a frog or a fish or an amphibian, of newt, cut the connections around the periphery that hold the eye in place, rotate it 180 degrees, and then sew it back in. And if he leaves the optic nerve intact, right, then that's basically the equivalent of having put a reversing prism in front of the eye. Everyone see that? And so the animal, as soon as it came out of anesthesia, it could make use, it could clearly see in that eye, as you'd expect, but its behavior let you know that it was, in fact, getting information wrong. If you held a tasty worm up, it would throw its tongue down to try to get it. If you held the tasty worm to the right of the eye, it would send its tongue to the left, etc. And to anticipate Justin's question, some of the animals, 4 months later, they still could not make use of that information in that eye. So apparently they don't have higher centers that can recalibrate how the sensory information is being used, and etc. And then the other,
Don't put it, he covered one eye, put the fix it.
Oh.
Stopped those very after 2 days. You were to close your eye with the fizzer and look through the eye that was covered over.
Yeah. I don't know that he ever did that. I mean, he assumed that there'd be a big discrepancy between the two eyes, and that was what he couldn't make sense of it at all. And that was why he couldn't work out the optics right to have the reversing prisms that the fellow in the 1930 photograph had. So yeah, I don't know. I mean, presumably the discrepancy was just too confusing that he couldn't make sense of it. So frogs apparently do not have any higher centers that can recalibrate the information coming from the eye that is now getting its information reversed. The interesting thing that happened is that when, in addition to cutting the eye loose, he also cut the optic nerve, rotate the eye 180 degrees. Of course, the eye's blind at first, but in frogs, unlike us, the retinal ganglion cells will regenerate, and eventually he could show that there were connections from the eye to the brain, and the animal's behavior indicated that those connections were the original connections. That is, because the animal's behavior was still confused about up, down, left, right, he concluded that the retinal ganglion cells had reestablished their original connections in the tectum. And the poor frog, without any higher visual centers to recalibrate, continues to show that, mistaken interpretation of the visual world for months. So far, so good? And Sperry, as we talked about before, he offered the chemoaffinity hypothesis, this idea that every retinal ganglion cell has a particular identity and will only innervate a particular tectal cell. And so that when they grow back, and presumably this is something that happened during development too, when they grow in, they go to the right place because each one's got a key that will only fit one particular lock. And one version, it could be, you could imagine that, every individual neuron has a distinct identity, but it could be as simple as just two gradients. Imagine one of these gradients, for example, are effrins. And so it could be that if the retinal cells are looking for a particular density of blue gradient and the yellow gradient, that they would end up re-establishing the old connections. Things get confusing when you try to think about these experiments from the point of view of the visual field onto the retina. Okay? So here, trying to make it sensible, here's a frog whose left eye is seeing a great blue heron just about to gobble it up. This is life and death information. And in this case, dorsal retina, when things are as they should be, dorsal retina is seeing the legs, ventral retina is seeing the head, etc. And this is what the pattern looks like on the eye, and this is how it ends up being projected onto the tectum. So left eye to right tectum, and here's the entire tectal field, and there's a topographic map of the world onto the eye, and a topographic map of the retina onto the tectum. And when he rotated that image and cut, what he saw was the original pattern. The cells re-established their old connections. Why? Because, the top of the animal, pardon me. Yeah. That is, the red retina is still going to lateral, right? What used to be dorsal is now ventral, etc. And so it reestablished those connections. And it gets confusing pretty quickly. But, and therefore, its brain has a reversed idea of where the heron is, and that's really bad. And it could never manage to unlearn that. So, those experiments were originally interpreted as a monumental win of nature over nurture. Because even though the information that was getting to the brain was totally maladaptive, the animal could never learn to make use of it. And even though the information was maladaptive, the retinal ganglion cells still reestablished their old connections to the tectum. And so what will happen then in the 60s and 70s and 80s is various neuroscientists will try every possible variation of this experiment that you can imagine. In part, it was because frogs are pretty cheap, and also because the way these maps actually got worked out, nobody had the anatomical tools to do it, but you could do it physiologically, where you'd have a frog, anesthetize it, have a screen in front of it, show images on the screen, dots or whatever, and then have an electrode in the tectum and find one particular tectal cell, find out what part of the screen makes it fire, move your electrode to a new tectal cell, etc. And so in one animal prep, you could basically map out the visual field in an afternoon. The sort of thing that you couldn't do anatomically in those days. So there were lots of variations of this. And the first take-home message is that the pattern of retina onto the tectum was always reestablished. And that was just be what Sperry's chemoaffinity hypothesis would suggest.
Yeah.
What was interesting, though, is it eventually did experiments like this, where they do the rotation, and now they remove half of the retina. So there's only half of the retina that's there. And the question was, will the remaining half of the retina, will it innervate only the part of the tectum it originally innervated and leaving the rest of the tectum naked, or not? And the answer was not. That even though it was half of the retina, it projected to the entire tectum got input. Even though, right, looking up here, this part of the tectum should have been listening to another set of retinal ganglion cells. And so it was, and yet the overall orientation of the field was preserved. So this sort of thing was referred to as a tectal expansion. Somehow the remaining retinal ganglion cells expanded their input. And so now every retinal ganglion cell was inputting to more tectal cells than it used to. And yet the overall topography was preserved. Or people would do experiments like this where they rotate the eye, cut the nerve, etc., and now they delete half the tectum. And so does that mean that the part of the retina that would have innervated that missing tectum, does that mean those axons don't make any connections at all? No. In this case, the retinal tectal projection was compressed so that every retinal ganglion cell still had some tectal cells to talk to. And yet again, the topography was still preserved.
They're still rotating for eye with this.
Pardon?
They still rotate for eyes with the together.
They would do these experiments typically with one eye. Is that what
Are they still rotating it?
Yes, absolutely. Yes. Yep. Yes. And although, I mean, once you map out the field, it doesn't matter. But, and, pardon?
It's not very.
Let's see. So, okay, you're right. And as depicted here, I mean, believe me, every permutation was tried, but as depicted here, they didn't do the rotation. But I see, I don't know why the scissors are missing here because, yeah. Although, as we'll see in a moment, that would happen. That sort of tectal compression will happen whether you cut the optic nerve or not. So there's no way to reconcile this with the strictest version of chemoaffinity that Sperry talked about, right? Because if the brown cell was only going to innervate brown tectum, then when you got rid of the brown cell in the tectum, it shouldn't have projected anybody. And yet the fact that the overall topography was preserved suggests that there's still, some order there. And so once people found the tectal compression and expansion experiments, they realized there was something about the life history of frogs that meant that the strict version of chemoaffinity would never have worked. Frogs, unlike mammals, continue to grow, right? The older the frog, the bigger it is. And as it grows, its eye grows. And as its eye grows, it needs more receptor cells. So in fact, each year, the frog adds a new layer of receptors and retinal ganglion neurons to the periphery of the eye. That's just how it grows. And another way that they're more plastic than we are, right? In that and it could be that this is why they have such plasticity in these systems, is that they need to keep adding retinal ganglion cells. Anyway, so they're added to periphery, and that means that the tectum is also growing. There's neurogenesis in the tectum too in adult frogs, but the additional neurons are always added to the posterior end. So the only way the topography would be preserved as the retina grows in one direction and the tectum in another is that there's got to be this constant shifting of inputs from the eye to the tectum that's going on. And yeah, and that too is inconsistent with a strict chemoaffinity hypothesis. Oh, and this is to remind you that in frogs, there's almost complete crossing of the midline, right? So in prey species like frogs, rabbits, etc., the eyes are oriented outward so that you have the biggest possible visual field to look out for predators, whereas in predatory species like cats and monkeys and humans, the eyes face forward because that gives us additional depth perception cues. And yeah, I guess that's why you have that there. And I told you every permutation was tried. Here was perhaps the bizarrest, which is folks made a frog that had never been made before that had 3 eyes. Take the primordial eye from a tadpole, graft it onto the head of another tadpole, and in some cases, the 3rd eye will get its axons to the tectum. And so now, even though normally each tectum receives input from only one eye, or primarily only one eye, now each tectum will be getting input from one tectum, will be getting input from 2 eyes. And what they found was that instead of the 2 eyes each homogeneously reproducing the map, the topographic map, they were taking turns. That is, one eye would be innervating one portion of the tectum, and the other eye would be innervating the others. And within each of those retinas, the overall projection was true to the original. And it turns out that this segregation of inputs in the 3-eyed frogs is visual experience dependent. If you silence one eye so that it can't, if you silence one eye, I'm not showing it here. If you silence one eye with TTX, then it loses out and the other eye takes over the tectum. If you put the animal in a situation where the only light it gets is a strobe light, and in a dark room with a strobe light, every time light happens, all of the retinal ganglion cells fire at once. If you did that, no segregation takes place. If you silence both eyes, no segregation takes place. So it looks like in these 3-eyed frogs, the 2 eyes are competing for space in a Hebbian-like fashion in the tectum. And that competition is being mediated by NMDA receptors, because when you block NMDA receptors, the segregation doesn't take place. And here's the thing that no one understands. These stripes of input from the 2 eyes in the weird 3-eyed frogs, they're always oriented rostrocaudally. Don't know why. And here's the other thing that I think is kind of strange. The width of these stripes is about 0.5 millimeter wide. And nobody knows why that is either. And I don't think anyone's making any 3-eyed frogs anymore. Right. The point was made. So what was originally presented as proof that nature was more important than nurture, it turned out that it was kind of true and kind of not. Nature, the lack of a visual experience at least, will get the retinal ganglion cells to the approximately correct part of the tectum. And once that happens, then visual experience will tighten up the focus of those inputs so that eventually you have cells that are next to each other in the retina tend to be ganging up on and driving cells that are neighboring each other in the tectum. And with that system in place, and presumably, apparently it's there throughout the life of the frog, that means that as the eye keeps growing by adding neurons to the periphery, and the tectum grows by adding neurons to the posterior portion, still there will always be that topographic map of visual field onto the tectum. And for it to work effectively, even in frogs, there's got to be a period where there's visual experience to tighten the input. And the other way people would look at this is to record the receptive field and record which parts of the tectum respond to which parts of the retina. And if there's been no visual experience to tighten those fields, those maps are very, very fuzzy and blobby. But with visual experience, they get tightened, sort of like when an image is first out of focus, and then you change the dimensions of the system and they come into focus. And so that experience is necessary for a good map. Nobody tried to probe how important it is for the behavior of frogs, but next we're going to talk about experiments looking in mammals, and it turns out that visual experience is also crucial for constructing the visual system in mammals, and in mammals at least, without visual experience, the system won't work at all. The animal won't be able to make use of the visual information. Questions about that? Oh, let's go for it. Let's have another go.